A major problem in our world today is that the overuse of antibiotics has decreased their effectiveness. To combat this problem in the future, we will need to come up with new methods of treating bacterial infections. One possible mode of attack is targeting the infection pathway. Current antibiotics rely largely on disrupting essential processes of the bacteria. As a result, there is high pressure on the bacteria to develop resistance to antibiotics, as non-resistant bacteria will then be killed by the antibiotics. If we can develop therapeutic agents that target a non-essential process such as the infection pathway, these agents will be less prone to inducing bacterial resistance. However, before we can develop new methods of treating bacterial infections, we need to fully understand the infection pathway. A crucial step in bacterial infection is adhesion to cells, a process mediated in uropathogenic E. coli by fibrous surface organelles known as pili. In my study, I examined the process by which E. coli assemble and secrete these pili. One protein involved in the biogenesis of pili is an outer membrane Usher. Previous studies had revealed information about the mechanisms of the Usher, but had never before explained the mechanism of its catalytic activity. To examine the protein interactions of the Usher in vivo, I modified a technique called in vivo site-directed photocross-linking, which had not been previously applied to this system, for use in studying the Chaperone Usher system of Pilus biogenesis. Using this technique, I gained a better understanding of its mode of action. To do this, I used three major steps. First, I created mutants that could incorporate a photocross-linkable amino acid. Second, I tested these mutants for proper expression and folding in vivo. Third, I cross-linked the Usher in the presence of Pilus tip subunits and analyzed the cross-link compounds to determine the interactions in vivo. Based on my results, I found that the disulfide loop region of the Usher's N-terminal domain serves a structural purpose in catalyzing subunit-subunit interactions, thereby catalyzing Pilus assembly. By contributing to the understanding of Pilus biogenesis, my project has put us one step closer to developing better treatments for bacterial infections.